It's my pleasure today to introduce Gerit Bethke, who's a postdoc in my lab. Garrett obtained her PhD from the Leibniz Institute of Plant Biochemistry in Halle, Germany, and she worked with Georg Schiele, and during that time she studied Arabidopsis MAP kinase signaling and plant immunity in botrytis and Pseudomonas syringae. Her first postdoc here was at the University of Minnesota with Jane Glazebrook, where she studied the role of cell walls in Arabidopsis, Pseudomonas syringae, botrytis, and alternaria interactions. She then came to my lab and she's been working on some Fusarium head blight work, which she'll talk about today. But she's currently working with me and Brian Stevenson on a relatively recently developed approach to rapidly isolate disease resistance genes in crop plants. For this project, she's working on barley and has a large set of resistance genes that she has in the process of being cloned, and hopefully within the next year or so we'll have them functionally characterized. And so this is an exciting project that's not quite developed enough to talk about it today. And so she's going to talk about some work that she did in my lab, she's done in my lab on a particular barley gene, this UDP-Glycosyltransferase, and its role in conferring resistance to Fusarium head blight, as well as providing resistance to the toxin that the fungus makes during infection. And so with that, I'll turn it over to Garrett to present her work.
Thank you, Gary. Yes, thank you for the opportunity to present my research here. And I'm gonna go right in. And so, as Gary mentioned, I'm working on Fusarium head blight resistance in wheat and barley, or mostly in barley, what we're going to talk about. But Fusarium head blight is a serious disease, both on wheat and barley, and this is mostly in humid or semi-humid climates, as we have here in Minnesota. And it's caused by a fungal pathogen called Fusarium graminearum. One important part of the disease progression is the production of mycotoxins of the trichotesine family. And you'll hear me talk a lot about one particular member of that trichotesine mycotoxin family called Deoxynivalenol. And FHB resistance is partial and it's quantitatively inherited. So there's no one particular mechanism that can make all wheat or all barley resistant to head blight. And first I want to tell you a bit about the terminology, because generally the resistance to Fusarium head blight is considered to be a two major mechanisms involved there. One is the type 1 resistance to Fusarium head blight. That means that it's the initial ability of the plant to protect itself against initial infection with Fusarium graminearum. And the second is the type 2 resistance, which is the type of resistance that prevents the spread of FHB infection between different kernels on the barley spike. And so this is a barley spike here, and you have all the different seeds sitting on it. And generally, when you look at the susceptible wheat plant, they often lack type 2 resistance. And a plant that gets infected at a point, maybe here or here, doesn't have this type 2 resistance that protects from spread of the disease. And so the rest of the spike bleaches out and gets white and is dead. The fungus can grow on the dead tissue. On the other hand, in a susceptible barley plant, generally type 2 resistance is present. So individual kernels might be infected, as you can see here. But it doesn't spread through the entire spike, so there'll always be healthy kernels in between. And so these trichotesines, these toxins, are virulence factors in wheat. They're important for the virulence of the pathogen. These trichotesines, or deoxynivalenol here as an example, they are protein biosynthesis inhibitors. And when Proctor et al. Developed a strain of Fusarium head blight where they knocked out the first gene in the biosynthesis of these trichotesine mycotoxins, they produced a Fusarium strain that couldn't make any glycotesines anymore. And this one was much less susceptible on wheat plants compared to a wild type Fusarium graminearum strain. It causes strong disease in susceptible wheat. And another line of evidence that these mycotoxins are important for the function or for the disease progression of Fusarium, and that disruption of the production or detoxification of the toxins themselves can lead to stronger resistance is in FHB7. So there was a very interesting science paper from about a year ago that was found that FHB7 was acquired by a relative of wheatgrass, Phenopyrum ponticum, by a horizontal gene transfer from a fungus. And then the wheatgrass was crossed with wheat, and so wheat plants could be then generated that carried this FHB7 gene. These plants were much more resistant to Fusarium head blight versus wild-type wheat plants, which are much more susceptible. And so what happens here, what is the function of that gene? FHB7 is a glutathione S transferase that detoxifies the Fusarium-produced tricotesines by conjugating a glutathione group here onto the epoxy group here in red, and then hence detoxifying the mycotoxin in the process. Also, a few years ago, it was found that in Arabidopsis, a UDP-Glycosyltransferase called DOCT1 provides resistance to deoxynivalenol. And so what the enzyme does, it takes the deoxynivalenol toxin and adds a glucose group on the C3 atom here and produces DON3 glucoside. And the DON3 glucoside is less toxic than the deoxynivalenol. And so one of the experiments in the paper was they created lines where they constitutively expressed with a, under the 35S promoter. So the attacked version of DOCT1, which they could then track in the plants. And so constitutive expression means that at all times, a high level of the protein is made in the plant. And so if you have wild type plants that don't do that and put them on medium containing DON, then they died. But if they had plants that overexpressed this gene here, DOCT1, then these plants were much healthier, bigger, greener, and they could survive. Suggesting that the role of the enzyme in turning the deoxynivalenol to the DON3 glucoside plays an important role in the resistance of these plants to deoxynivalenol. So we also know that deoxynivalenol is converted to DON3G in barley. So this was work done in Gary's group in 2010. And they inoculated a barley spike, a susceptible barley line with deoxynivalenol, and looked at the time course experiment here. And you could see in black that the deoxynivalenol content in these plants went down over time versus the amount of deoxynivalenol 3 glucoside, the DON3G, which is the less toxic compound, went up. Gary's group was then interested also in finding out what genes might be responsible for that process. So they did 2 different experiments published in 2 different papers. One was that they used Fusarium graminearum wild type, which makes the deoxynivalenol, and the tri5 mutant Fusarium, which doesn't make any glycotesines, and looked at the expression of genes with a microarray experiment. So it looked at about 22,000 barley genes, and they looked for genes that were expressed only in wild type, but not or with wild-type Fusarium, but not with the Tri5 mutant. And so this is one way to find trichoticine or mycotoxin genes. Another method that was employed was where they inoculated the plants directly with the toxin, DON, or with water as a control. Looked at a similar experiment, looked for differentially expressed genes that were only induced after deoxynivalenol treatment, and also found here trichoticine response. Among these genes were quite a few UDP-Glycosyltransferases. In total, about 9 could be identified. And there's circa 100 UDP-Glycosyltransferases in barley, so it's quite a large amount of those that were induced by trichoticine or Fusarium infection. And to study the function in a bit more detail, 4 of those were then put into yeast, and the function compared to this Arabidopsis gene that I just talked about, the STOC-T1, which is the UDP-Glycosyltransferase from Arabidopsis, known to be able to convert DON to the DON3 gene. There's also a negative control here in this experiment, which is just an empty vector expressing no protein. And there were 4 barley UDP-Glycosyltransferases here. The first one is the most closest homologue to STOC-T1 in barley. The second one is UGT13248, which the majority of this talk will be about. And then these are 2 other UDP-Glycosyltransferases from barley. And you can see only the UGT13248 provided resistance to the yeast at increasing amounts of deoxynivalenol, at very comparable levels to the Arabidopsis STOC-T1 control. This is the Western blot showing that these proteins were made in yeast. And then also over time, in the supernatant with the UGT13248, a large quantity of DON3 glucoside could be detected, again suggesting that the UGT13248 could function in a conversion of deoxynivalenol, the mycotoxin, to the less toxic version DON3. And another postdoc in Gary's group then put this gene into Arabidopsis. So again, overexpressing it under the constitutive 35S promoter with a tag as a tag version of the protein. And you can see here, this is wild-type Arabidopsis, and it doesn't do particularly well on medium containing deoxynivalenol. So the plants stay very small, they bleach out and die at high concentrations. In comparison to that, Arabidopsis plants here are 3 independent lines that carry this gene and expresses constitutively. They are much better able to deal with, again here, increasing concentrations of deoxynivalenol. Another, this time it was a graduate student in Gary's lab, worked on to putting this UGT13248 yeast, actually a crop that gets infected by Fusarium graminearum. And you can see here, this is a time course experiment. And here in BW is Bob White, which is a wild-type beet. And you can see after infection or after treatment with deoxynivalenol, the amount of deoxynivalenol gets reduced over time. This is days after inoculation, versus the amount of DON3G goes up. So it happens even in wild-type barley, suggesting that the conversion from deoxynivalenol to DON3G happens even in the wild-type Bob White. But then these plants also overexpress the barley UGT13248 here. This is an example, line number 19. You can see the same process happens, but much, much faster. So this is the deoxynivalenol, which is reduced very early on in infection. In 3 days, it's basically at the low level, versus the DON3 glucoside, which increases also rapidly. And this figure shows the same experiment, but shows a ratio of the DON3 glucoside to the deoxynivalenol. And you can see here the plants overexpressing have much more DON3G compared to DON as in comparison to the wild type in yellow. And those plants turned also out to be resistant to Fusarium graminearum. So what Chin did here in 3 different experiments, he inoculated 2 spikelets on either side of the spike of wheat with Fusarium graminearum that makes deoxynivalenol. And you can see here the plants overexpressing the UGT13248 at much less disease severity as compared to the untransformed wild type or white. And there's also 2 controls here. Sumai3 is a resistant genotype, and Wheaton is a susceptible genotype. Interestingly, as I said earlier, Fusarium head blight is a quantitative trait. And so various phenotypes have been associated with resistance to Fusarium head blight, one of them being height, or heading date, or grain protein content. But it's interestingly enough, none of those QTLs hits the position of where UGT13248 is present on chromosome 5 in Barley. Yedong Huang in Gary's group has also found, when he looked at near isogenic lines that carried with in one year resistance found on chromosome 2H of Barley, that the resistant lines early on in infection expressed the UGT13248 at higher levels than susceptible plants. That was also true for near-isogenic lines that carried a resistance that he had mapped to chromosome 6H early time points. We don't know why that is because, as I said, the UGT13248 is located on chromosome 5. It is possible that those would be regulatory proteins that regulate the expression of the UGT13248. Yedong then also sequenced a set of 28 Barley genotype with known resistance phenotypes. So in green are moderately resistant or resistant genotypes, and in red are moderately susceptible or susceptible genotypes. And he found 4 mutations in UGT13248 in plants. This was to find out, what possible variation is present in the various genotypes, and could those be utilized to understand the role of UDP-13248, UDP-Glycosyltransferase 13248 in Barley. And so 4 mutations were found, one at position amino acid 20, one at amino acid 50, one at 105, and the last one at position 213. And this is a schematic overview of the UGT13248 protein. It has 3 exons, exon 1, exon 2, and exon 3. In this region here is a domain called the PSPG box, which was early on associated with the active site of the protein. And so the mutations that Yedong found in those 28 Barley genotypes were all on exon 1, which is not close to the proposed active site. We then also worked with Stella Wyand in Peter Morell's group, and she looked at exome capture sequence data that basically covered the second and third exon of the UGT. And the data that they had were for 34 elite Barley lines, 132 wild Barley accessions, and 317 land races. And Skylar was able to identify 7 mutations, and those were present in less than 2% of the 533 total genotypes of elite lions, wild barleys, and races. So we think this is fairly rare. And also we looked at genotypes that carry any of these mutations and put seedlings on plates containing mock control or deoxynivalenol, and we didn't see any differences in the sensitivity to deoxynivalenol on plants containing any mutations in there. I think none of these mutations has an effect on the function of the gene. So now, in order to study the function of the UGT13248 in barley, we use two approaches. So the first one is shown here. So we created, again, overexpression lines. So in this case, this is the construct. So instead of the 35S promoter, which is constitutive promoter that works in our boxes, we use the ubiquitin-1 promoter from corn that works in barley, and transformed a construct of the UGT13248 with a flag tag under the control of this ubiquitous promoter here. We found multiple independent events. This is a Savon plot showing the presence of a construct that can be probed with a UGT13248 specific sequence. And you can see here that full-length genomic DNA was digested, and the patterns of where these probes bind are different for these different lines, suggesting that these are independent insertions into the genome of golden promise. So this should say golden promise and not Bob White. I apologize. And this is a Western blot analysis showing that this is the wild type control, but the three plants contain the protein which was targeted with this tag that was inserted in the transgene. And so the first experiment we did with this was, and so this was done by Xin Li. He prepared medium containing no deoxynivalenol or 5 parts per million deoxynivalenol. And the golden promise plants that were put on medium containing no deoxynivalenol grew to a certain length of time. And at that same time frame, these wild type plants did not grow as much in medium containing deoxynivalenol, suggesting that those plants are susceptible to deoxynivalenol. However, when he compared these overexpression plants here, so these are the 3 different lines, then you can see a much longer growth compared to the wild type in these genotypes, suggesting that overexpression of the UGT13248 makes those plants less susceptible to the toxin deoxynivalenol. And here's a quantification of this. So this is UGT minus are plants that do not express the UGT. And you can see here in a mock treated plant, this is the root growth. So this is the root length over time, and this is untreated. And then when those plants that do not express the UGT are treated with deoxynivalenol, there's a reduction in root growth. And this does not happen in plants that carry the overexpression construct for UGT 13248. Those plants here are indistinguishable in root length between mock and deoxynivalenol treated plants, so suggesting that those plants get more resistant to deoxynivalenol. And when we look at the data here over time, and we inoculate the toxin deoxynivalenol directly into the spike of barley, then we can see that more DON3G is produced faster in the lines carrying the UGT overexpression construct compared to lines that do not carry it here. So we have a similar effect that we found before in wheat, also when we overexpress UGT13248. barley. So we conclude from this, so if we overexpress the construct, we produce a large amount of protein, and the plants that produce this large amount of protein convert more of the toxin deoxynivalenol to the less toxic form, deoxynivalenol 3-glucoside, and these plants are more resistant to deoxynivalenol. Next, we decided to utilize a different method where we were looking at mutants. Mutants would be plants that have an impaired function of the enzyme UDP-8. And to find these mutants, we utilized a tilling population. Those Pomorex seeds that had been mutagenized with sodium azide in Silvio Salvi's lab in Italy. You grew the M2 and the M1 and M2 plants up, and then identified in the M3 4,906 different families, extracted genomic DNA from that, and then scanned for mutations in a construct that covered this active site PSPG box in the UGT-13248. So we don't make primers for this. And then we got 4 lines back from Silvio's group, and we made sure that they actually contained the mutations found. And so with that, we identified 3 lines that had a change in the protein sequence. So there were mutants at the position 368 of the protein, 369, and 43. And we also identified 1 silent mutation. So there was a mutation in the nucleotide sequence, which did not lead to a change in the protein sequence. And we call that one Stillmore 999. And here is an overview of the mutations that we found. So previous to this work, a very close relative or homologue of UGT-13248 in rice was crystallized, and a crystal structure of the protein was made. And they found, and they crystallized it together with, here in purple, triclotesine and the substrate. So the substrate of the enzyme is a UDP glucose molecule, which then gets put onto the triclotesine to make a triclotesine 3-glucoside. And the active site they found is a threonine at position 299, which has an interaction with the substrate, the UDP glucose. And the other active site was this histidine 38, which interacts with the triclotesine in this publication. So in wild type version of the Barley UGT-13248, these amino acids here, the histidine 369 and the threonine 368, show hydrogen bonds with the UDP glucose. In the mutant versions here, we found the threonine 368 isoleucine version, where the threonine in position 368 was changed against an isoleucine. We lose one of those hydrogen bonds. And in a version, in the other mutant, the histidine 369 tyrosine mutation here, all 3 hydrogen bonds that you can see in the wild type are lost. And so likely those proteins will have an effect on the substrate binding of the UGT-13248 and possibly on the function. And we also found another mutation, serine 403N, but this one is further away from the active site and less likely to have an effect on the protein function. And so indeed, when we did the same assay, as I showed you, plate assay, where we grow seedlings on medium containing no deoxynivalenol, or in this case, 1 parts per million deoxynivalenol. we can see that here, that, for example, Morex, that is the wild type, you see a slightly longer root growth without the, actually you can't see a difference between root growth because the error bars overlap between Morex grown on deoxynivalenol containing medium and MOC containing medium. But when you take the H369Y mutant here and look at the MOC treated plants, they have the root length is not distinguishable from the wild type plants. But if those plants get treated with deoxynivalenol here in the light green, those plants are severely stunted. So they're much more sensitive to the DON, or much less, which is the opposite effect of if you overexpress it, and then the plants get more resistant. And the same is true for this T68I plant here. Again, EMOC-treated plants of certain length. And then after deoxynivalenol treatment, they're much shorter, suggesting that they're hypersensitive to DON, possibly because they can't detoxify the DON to DON 3G. And to test this, I inoculated spikes of all these genotypes. So Morex is the wild type, this is the silent mutation, and this is the mutation that is far away from the active site. And here's the T68I mutation that loses one of these hydrogen bonds. And here's the H369Y mutation, where you see the loss of all 3 hydrogen bonds. And you can see those plants produce much less DON 3G compared to DON over time as the wild type. So suggesting that the function of the protein is severely reduced and that this is a stronger allele even than this allele. And so when we looked at FHB resistance in the field, here again we have Morex. We have plants that carry the T368Y mutation, plants that carry the H369Y mutation, plants with the far away mutation S4. And it doesn't have an effect on deoxynivalenol conjugation. These silent mutation plants that have no effect either. Susceptible control and a resistant control. And you can see the T368I and H369Y plants are much more susceptible than wild type or this mutation. This is far away. And this we've done this in 2 years, 2019 and 2020, and we've done it in 2 different environments each, St. Paul and Crookston. And you see basically the same effect, that these mutants are more susceptible compared to Morex in both environments. Oddly enough, the tilling line with the silent mutation was also very susceptible, but we think in this case this is due to the fact that they're also very short. And height has been found to be a factor in FHB resistance in the field. And also in the field, we see the same phenotype as described earlier, that these, so in this case, we're looking at the DON and the DON 3G production after infecting the plants with Fusarium that can make deoxynivalenol. And we can see here that these T68I and H369Y mutants make much less DON 3G compared to DON than the control. And so these controls are again Morex wild type, the tilling line, which has a silent mutation, and also plant with a mutation far away from the active site. And so then we wondered, okay, what other effect might this UGT have on FHB resistance? And I did a point inoculation experiment where we where I put FHB in 2 spikelets, 1 on each side of the spike, and then tracked disease severity. So here you can see again Morex is a control, the wild type plants. And you see there is not much increase in FHB severity over time. So 7 DPI is when we see the first symptoms, really, up to 14 days after infection. And you can see the T68I and the H369Y plants are already more susceptible at early time points, but this susceptibility increases over time. The S403N plants are similar to wild-type Morex, and those short Tilmor 999 plants are a little bit more susceptible, but not at the level that you see for the mutant plants that have a mutation in the UGT13-2 protein. And interesting when you look at the phenotypes of these T368I and H369Y plants, they're more similar to what I showed you in the introduction in wheat. So the spike basically dies above the inoculation site. There's some bleaching, and there's a spread of infection over time. From this, we concluded that UGT-13248 is required for type 2 resistance in barley. And so the conclusions from the mutant experiments here are that the UGT-13248 that has mutations in T68I and H369Y position, those plants can convert much less toxic DON to the less toxic DON3G. Those mutant plants are also more susceptible to Fusarium head blight, and they have reduced type 2 resistance to Fusarium head blight. Summary of the entire talk would be that the UGT-13248 does conjugate DON to DON3G in barley, as previously shown in various other plants and in yeast. The mutations in the UGT-13248 are very rare, mostly only found in wild barley and a couple of land races. And we have not been able to identify any mutations that affect function. So likely this is an important gene that doesn't show a lot of variation in the body. And the UGT-13248 affects type 2 resistance to Fusarium head blight, and this might be the reason why we don't see many mutations in the UGT genotypes tested. And so for acknowledgements, I want to thank Gary Muehlbauer for allowing me to do this work and supporting this work in his lab. Jane Heinen is Gary's technician who is heavily involved in all the projects in the lab and helps with growing plants and process and stuff like that. Yedong Huang has worked with me on this project, so various of the findings were his, and Chin Lee and Shin were previous members of Gary's lab that has also done some preliminary work on the project. And at the University of Minnesota, we worked with Ruth Delmaki to look at disease resistance in transgenic plants. And Yang Hong has helped with deoxynivalenol measurements. Peter Morel and Skylar have been helping us with the exome capture sequencing analysis. And a lot of the genotypes we worked on came from Brian and Stephenson and Kevin Smith. And Yedong has also worked with Kevin's group to do the FHB test field. Gerhard Adams and Wolfgang Schweiger have worked with Gary before to look at the function of the UGTs in yeast, and Franz has been really helpful in doing all the DON3G and DON measurements where I showed both of them. We've got a lot of the deoxynivalenol from Susan McCormick over the years, and Jochen Kumlein and Götz Hensel have made the transgenic barley plants at IPK in Gartersleben, and Silvio Salvo's group in the University of Bologna in Italy have developed these tilling populations that we mutants in. And funding was over the years provided by the US and Barley SCAP Initiative, USDA, and the University of Minnesota. I'd be happy to take any questions.
So are there any questions for Garrett? So you can just put them in the chat, or I think you can probably arrange for you to talk directly to her if you want to. So I have a question. So what's the so when DON is converted to DON 3 glucoside, and then say someone eats that DON 3 glucoside, is that actually safe to eat that compound, or does it cause a problem? I guess my question is, does it convert back to DON in your gut or anything like that?
It does convert back to DON in your gut, but from what I understand, it's still less toxic. It might be after the point where it inhibits your, protein biosynthesis much more. Of course, the FHB7 type resistance with the glutathione transfer is non-reversible, but the glucose addition is reversible.
I think Jerry has a question.
Yeah, I would just love glucosyltransferases. So I have a couple of questions. So the first one is there a native substrate for this? I mean, the fact that you don't find very many mutants suggests that it has a function outside of disease resistance. And glucosyltransferases are, a little bit fickle in terms of compounds that they work on. So the question is, what is its, normal role outside of protection against infection? And then, okay, and then the second one, and then I'll shut up and you can talk. The second one is glucosyltransferases are nice because you can engineer them, and it would be really nice to make something other than a glucoside. If you could make an arabinoside or a galactoside, there are many fewer galactosidases and arabinosidases, both in the gut and in the plant. So you might get a much more durable, stronger inhibition if you made a different kind of sugar. So those are the two things. So I'll be
to the second one, that sounds like an interesting approach, right? So something that we could work on in the future. And it sounds, yeah, we've also tried to find glutathione S transferases, but we didn't nail one down that had a specific function in barley. So yes, having the ability to add a different sugar from glucose, which could less likely to be cleaved, would be a great, progress. And so as for the function, so there's about 100 UGTs, about 10 of them we looked at for the deoxynivalenol conjugation. And so, yeah, the one that we found to have the only one that had an effect on the deoxynivalenol was this UGT-13248. And we also found that that acts on various other trichoticines. So it's not really specific to the deoxynivalenol. And I know that UGTs play a role in like cell wall biosynthesis, etc. So in very many processes. We do not know if it has any other particular function, or what groups of UGTs specifically in barley have which specific function. I know from my work in Arabidopsis and cell wall related enzymes that sometimes the expression can give you a clue about what they function in, like pollen tube growth versus disease resistance, but we haven't done, to my knowledge, that sort of analysis in with the barley UGTs.
It would be a fun metabolomics analysis because you could just look for glucosides and ones that are missing in the
Yeah, it's true. Now that we have the mutants,
Yeah.
maybe we should check in with you or Adrian and see what could be done.
Well, it might be nice in terms of engineering resistance if you knew
Mhm.
what kind of substrates are also being worked on by that enzyme. So
Yeah.
just a thought. But I think, if you focused on glucosides, or you could even, for example, feed in a carbon-13 label precursor or something,
Mhm.
and then look for changes between the different lines you have, would be useful.
Sounds like a really good idea.
So Jerry, the Arabidopsis gene conjugates steroids with a glucoside group.
Mm.
So I assume that's probably its primary role in Arabidopsis. The Barley gene that Garrett discussed today is not the most homologous gene to the
Only like 23% homology or something.
And so the gene that shows high homology to the Arabidopsis gene did not actually have a function with relation to deoxynivalenol. So it's likely that these genes are probably involved in things like that, like steroids or whatever. But when we overexpress the gene in both barley and wheat, we don't see a reduction in height like they did in Arabidopsis. The plants did not bolt at all. They were just they were basically a rosette formation, and that was it.
Well, that makes sense.
Well, it's interesting to hear, this conversation about engineering resistance. So what is the status of engineering of wheat and barley in general in the world right now? Where does it stand? Are there groups that are trying to move forward with genetic engineering or using CRISPR to bring some of these discoveries into practice in wheat and barley?
I think there are other people on this talk who probably know more about it, but from what I understand right now, there is a big reluctance to do anything to wheat because wheat is this holy grail and nobody wants to do any genome engineering on wheat. With barley, it might be a slightly different story.
Barley is probably even more conservative with regards to genetic engineering. So the malting and brewing companies don't want to have a PR problem associated with GMO crops.
Yeah, so no progress in that conversation has been made over the last 25 years, huh? No progress at all?
I would assume that what will happen is that China will probably release something that is GMO at some point. I don't know what that trait will be, but I assume that's probably where it will occur first.
Yeah, and from what I understand from the growers, when we went to the Wheat and Barley Initiative meeting, they really don't want to have transgenic wheat, at least, because they do a lot of export. And, as even the CRISPR stuff is considered GMO in the European Union, they're very hesitant to have any potential contamination.
Yeah, I'm well aware of that, but I was just wondering whether there is any consortium in barley and wheat and maybe other grains of the world to try and work through that roadblock.
I don't know.
Not that I know of. I mean, I think there are groups that work on GMOs in general, but not specific to barley and wheat.
Yeah.
Sorry, I was muted there. If there are no other questions, let's give Garrett a round of applause and thank her for her nice presentation.
Garrett, this is Don Wise. Would you mind staying on for a couple minutes after?
Yeah, no, yeah, of course.
Okay. Garrett, I've worked on Pseudomonas syringae over the years. I'm the guy that, well, my team, I didn't do it, isolated Pseudomonas syringae from Canada thistle, right? You know, Canada thistle populations, you know, it's a major perennial weed here in the upper Midwest and, you know, really around the world. And we discovered that there's this natural infection that occurs that turns the Canada thistle plant white. So we went in and isolated the organism, and it was Pseudomonas syringae. We then had the great idea that this was a potential biocontrol, and actually developed the technology to actually apply Pseudomonas syringae in a solution with a surfactant, and we're able to get infection, right? But this is what happened. We would see initiation of symptoms, and then the Canada thistle would recover within, the tissue would recover in just a few days. While in nature, once a plant is infected, it will continue, the symptoms continue to develop over a much longer period of time. So we did some mixtures and things, but to try and figure out how we could continue the expression of that toxin over a longer period of time. Now this goes back probably 15 years. This is the biggest failure I had in my career. that we were never able to figure out how to continue to have that gene express tegetotoxin in the Canada thistle plant. And I was just joking with some folks the other day that I might just stay around for another 5 years if I could figure out. I'm just kidding, Gary. If we could figure out, and I would have to go back and we'd have to do the due diligence. That's really what I wanted to ask you. What has been done in the last number of years in understanding the expression of the gene that gives rise to tegetotoxin in plants just in general? Has there been a lot of effort? If we could figure out how in the hell to do that, we would have a major, the organic farmers in the country, it's just a disaster. Canada is just a disaster, right? So they were always very excited to work with me to develop this capacity to use this bacteria to control Canada thistle in organic systems. And certainly would be other groups that would be interested in it. So when I heard you talk here today about your background in Pseudomonas, I was just wondering if there's been major advances made in understanding the production and what controls the production of tegetotoxin in a system.
I have no experience with that. So the Arabidopsis, you know, I worked on Arabidopsis diana and Pseudomonas syringae, and we looked at like basic immune responses, like, you know, the recognition of the pathogen,
I
that
see.
sort of stuff. So I have no experience with the toxin side of it.
I understand. Yeah.
You just have to figure out how to induce the toxin, huh?
That's what it is. That's right, Gary.
Why would it be induced in nature, but not in when you apply it to the plant? We
could not figure that out, but we weren't doing the basic, you know.
Could it be environmental factors like UV, drought, that sort of stuff?
Well, I don't think, it was across a wide range of environments and we would get the same response every time, right? But I would love to put a team together to go after that again. That's been, 15 to 20 years, and certainly technologies are there to actually go after it in a real concerted way. That I think it has the potential really to become a real potential biocontrol for that major species around the world, if we could figure that out. So Gary, you're going to help me?
Sure. No, Garrett will help you. I mean, it sounds to me like, so for example, that toxin that Garrett has talked about today, trichothecenes.
Yeah.
Those are not produced on just standard media. They're only produced when you there's a couple of different compounds, I can't remember their names right now, that actually induce those compounds. And then in plants, they don't actually become induced until the fungus gets to the rachis node of wheat or barley.
Yeah.
And so that there's some compound there that's telling the Fusarium that make toxins. And so if you go and isolate Fusarium from all sorts of native grasses around the state, no toxin production.
Yeah. Yeah.
So it's like, and the Fusarium is there. It's infecting it, but there's no toxin production, none.
Yeah.
So it's interesting that you can actually, in a sort of an agricultural setting, it's induced.
Yeah.
Plants have been bred in such a way that it allows that induction and susceptibility
Yeah,
to.
so we went through with all kinds of mixtures of things, and in terms of combinations. And I also think there's, the potential that it's not just the interaction of one microbe. There may be additional microbes in the plant that work together to produce that tox. I mean, but just pulling back from that, if we could figure out how to get that toxin to be expressed in that plant, it would be, a big breakthrough in biocontrol of Canada thistle. So that's what I wanted to just. I was just thinking maybe you knew a little bit about that in terms of.
I'm sorry, no.
Okay,
so I'm going to retire now.
All right. See you later, Don.
All right. Thanks, Garrett, for your presentation. It was really well done. Thank you.
Thank you very much.
All right.